Hi Irene, thank you very much for a very entertaining talk.
Thank you.
So, it is a very sophisticated design. You have two different settings, and can you tell us a little bit more about the design that you took on this research?
Well, the idea came from the possibilities that nature offers us. So since there are natural CO2 bands, which are locations that for long has been exposed to high CO2 levels, we decide to take advantage of that and go there and then select locations which are under the direct exposure of CO2 and another closer but not under the CO2 volcanic influence. That will be the reference one. And then we perform incubations there. We collect the plants and we incubate it in closed chambers that were gas tight, and we measure the oxygen evolution as a proxy of the productivity. And we also took samples for gene expression. That was the combined approach we took for linking at different levels between gene expression and productivity. This approach allows us to estimate the long-term effects that ocean acidification would have on seagrasses. But then we were also curious to see the short-term response of plants and also the reversibility capacity of these plants to readapt or reacclimate better to the original or to contrasting conditions. So what we do was a kind of transplant experiment where we collect plants from the CO2 site and incubate it with water from the reference site, and otherwise plants from the reference site and incubate it in water from the CO2 site. And again, evaluate the response on productivity and gene expression.
And so the idea coming in was that higher CO2 would lead to higher productivity. It is the standard that people think. It's like almost common sense, shall we say it. And as any great scientific result, that was not what you saw.
Yeah, exactly. The original hypothesis that we were expecting somehow to confirm was that the input of CO2 into the system, into the sea, this increase in DIC would increase or would promote productivity. But instead what we consistently measure in the CO2 bands is that plants that were exposed to this CO2 water, both in long and short time scale, had lower productivity rates than the reference one, and also a consistent downregulation of the photosynthetic genes.
And what is the explanation that you have for that?
Well, after the first shocking response we had from the results, what we thought that was first we had to be sure about the robustness of our results. For that, we ran another set of incubations here in Ria Formosa, where we control all the physicochemical parameters, and we were sure that the only thing that we changed between incubations were the CO2 concentration. And then the results show that actually there was a significant correlation between net plant productivity and CO2. So we were sure that when we control everything and you only modify the CO2, the productivity of the plants increase. So something is going on in the bands where we were working, that the response was exactly the opposite, both in short and long-term exposure.
And that, of course, leads to the question, what are you going to do next? And I know that you have been awarded a highly competitive and very extraordinary fellowship, Madame Curie, so congratulations on that as well.
Thank you.
And are you going to follow the work that you just showed us with that fellowship?
Yeah, well, in some way, yeah, the idea is continue this line. We still have a lot of work here to carry on this, because I show the results, but we have a lot of more work that is still processing. So we hope to put more light on this issue. And of course, the Marie Curie will follow on this line, but try to widen a bit more, not only on the single effect of ocean acidification, but more on the multi-driver effect, the combined effect of ocean acidification, temperature, and other drivers associated to climate change, using seagrasses as a model species.
Okay, good luck with that, and thank you again for a wonderful talk.